Now that we have optimized our segmentation parameters for this representative dataset, it's now time to run some analysis. I know that I'm working with the single cell dataset, but if I was unsure what was available, I could navigate to the documentation by clicking on the Help window, scroll down, and once I hit the Analysis section, I get a preview of what kind of analysis modes are available to me. We can see that we have 5 options in the current release. The analysis mode, None, will simply segment our objects as we've configured them and save them. It will not conduct any analysis on the segmented objects that we have produced from our original images. If we select the basic mode, we can generate our 3 object types as per sort of our expectations from the optimization mode, but there are no relationships established between any of those objects. And all segmented objects have some basic measurements around them, but there are no relationships asserted between the object types. Then we have 3 biomodes: spheroid, single cell, and speckle. And we have fairly lengthy descriptions here of what each of the relationships are pertaining to those various modes. So for example, in spheroids, we have lots of nuclei related to a single spheroid. So we get a breakdown of which nuclei was inside each parent spheroid. And we also get some additional analysis in spheroid mode that calculates the number of nuclei inside that spheroid. We also get to know which nucleus belong to each spheroid at a single nuclei level. And when we talk about additional label processing, we also get to know where those nuclei are distributed throughout the spheroid in 3D space. If we're doing analysis on single cells, as we are in this demonstration, the relationships that are established are what we'd previously discussed. We want to relate each nucleus to its parent cell, and if we have configured some options to create a cytoplasm, we'd like that to be included in that relationship as well. So ideally, we'd like one nucleus to one cell. However, we also have included functionality to detect and manage cases of multinucleated cells. Again, this could be a segmentation error, or it could be a biologically relevant phenomenon that needs to be described. And finally, in speckle mode, we have options to configure a single nucleus and relate speckle types 1 and 2 to that parent nucleus, with a number of other subsequent analyses available. So let's have a look at how this works. Let's open up the Analyze panel. It looks somewhat similar to the Optimize panel, but we have a few different options around the periphery here. The central panels remain the same. We still need to configure segmentation parameters for our primary objects, and that includes specifying the channel, any background and then foreground filters, what segmentation method we'd like to use, and if relevant, the size or parameters for that method. And then of course we need to also include a threshold. Again, it would be very painful if I had to manually enter all of these values every single time I wanted to do some analysis. It would also limit my ability to automate my analysis with any kind of scripting. So in order to speed up that process, we can use our configuration file from the Optimize mode. Note how my images for analysis are the only things in this particular folder. In fact, I've made them their own folder. There's my optimization file, and my images are in their own subdirectory. So let's go ahead and load that configuration file. We now see that all of my segmentation parameters from our previous session have loaded into this one, which is great. I don't have to remember all those parameters, and it's far more reproducible. We also see that I've ticked this tertiary equals secondary minus primary button. Remember, our single cell analysis mode needs to have primary objects as nuclei, secondary objects as whole cells, and an optional tertiary object which will represent the cytoplasm. Now a cytoplasm is just the whole cell minus the shape of the nucleus. So in this case, we can easily generate that for each cell by saying that our tertiary label will be equal to secondary minus primary. If I had a particular stain that I wanted to represent the cytoplasm and I wanted to segment that, instead of ticking this option, I would select the tertiary segmentation, select the channel, and then configure the segmentation parameters for that independently. Of course, that can also be optimized in the optimization mode. But in this case, I want to automate that process. So let's just subtract our nuclei from our whole cells to get a cytoplasm. I then need to say where my data is. Let's select the input directory, which will be the images folder that just contains my 2 images for this example. If I would like to save all the outputs from this analysis mode to a separate directory, I can select one here, but you don't have to. If the output directory is blank, the outputs for the experiment will be saved to the same input directory. Let's select the analysis mode we'd like to run here. Again, we have 5 modes. We'd like to use the single cell mode. Notice when I select that option, I get this process labels feature down here. When I explore that, I can configure some additional analysis for secondary, tertiary, and primary objects, respectively. We'll talk about those in the next video. Great, I think we're nearly ready to go. So we have a directory with images, an analysis mode specified, and some segmentation parameters for each of our object types. Great. We have these text box in the analysis window as well. If you would like for every object in this experiment to reference a particular group for your experiment, you can enter a variable here. For example, maybe all of the images in my experiment folder here are from my control group, or maybe they're from a drug dose that you'd like to record, or maybe they're from a particular condition. If you'd like everything to be affiliated with a group for your subsequent analysis, you can enter that here. For this purpose, I might write test. If you would like to give names to each of your channels that you will quantify the intensity of inside all of your segmented objects, you can enter those here as well. If you leave these blank, your channels will be referred to as C1, C2, C3, and C4 if it's available, respectively. Let's enter some variables so we can see what that looks like in the data output. I use DAPI to stain the nuclei in channel 1, and then we have channel 2 as a protein of interest, and channel 3 will be actin. If I'd like to, I can also add some kill borders options here. Selecting XY will remove objects that touch the X and the Y border, and obviously selecting X, Y, and Z will remove objects that are touching any boundary of my image. Let's just run with no for now. Cool, I think we're ready to go. When I click run, we'll see a number of files appear in our output directory. We get lots of information in the log. You'll also notice that as soon as I hit run, we had a FOCUST parameter file that was saved to our output directory. That's just like the FOCUST optimized file, except it contains all the additional information about this particular experiment that we're running right now, such as the variable names, how I generated my tertiary objects, my options for kill borders, the analysis mode, and if I'd selected any of our additional label processing, that would be there as well. You can see now that our FOCUST batch processing has finished, so we processed 2 images in 26 seconds. Cool. We could go back through and have a look at all the components of those logs, and we can see that at various points we did some segmentation, we analyzed those objects, we measured the intensity of each of the channels inside those object spaces, and we did a number of other things. Let's close this and have a look at what we have in this folder. We have a copy of our log, the parameter file, and then lots of very long worded titled images, as well as a CSV file. Our 2 original images are also in this folder. We can see that single cells 1, we now have primary objects for single cells 1 and single cells 2, secondary objects for single cells 1 and 2, the tertiary objects for each of our original images, and then these relabeled single cell images. We'll talk about those shortly. Let's open up the primary objects for single cell 1. They are indeed the nuclei from our single cell 1 image. Great. Let's have a look at the tertiary objects for single cells 1, which remember should be the whole cell shape minus the nuclei. Cool. We can see we have some whole cell shapes, and they're definitely missing their nuclei. So this is a cytoplasmic label. Let's have a look at the data. You'll notice that as I expand some of these headers, we've captured the individual name of each of our files. And as we scroll down, we go from single cells 1 to single cells 2. So we were able to batch process 2 images in a single experiment without us needing to write any code, which is pretty handy. We can also see that grouping variable that we added into our data set here. Group is test. Then we can see the label of the cells. And as I start to expand these data columns, we see that we get some interesting naming conventions. The important thing to note with the data set for single cell is that every single row represents a single cell. So what we've done in single cell mode is concatenate and match all the data together so that a nucleus and its cell and its cytoplasm are all inside of one row. So for example, label 5, primary.volume. This is my calibrated volume for the nucleus of cell 5. As I work my way across, I could go ahead and explore some of these morphological measurements as well. I could have a look at the mean expression of DAPI inside of nucleus for cell 5. As I keep moving across, we run into secondary object components to cell 5. For example, volume. We now have the calibrated volume for the cell of nucleus or cell 5. As we keep moving across, we run into the tertiary object. And again, the same row pertains to the same cell. This is the tertiary stuff for cell 5. We can also then have a look and double check that we've got some intensity values in this data set. For example, where is it? There it is. We can see the mean intensity of DAPI inside our secondary image. And of course, the DAPI is there because we entered it as a variable inside our channel name. If we didn't enter DAPI, that would read secondary C1 standard deviation. So we have a lot of data to play with here. Something I want to draw your attention to though is our label column. Normally we'd expect labels for segmented objects to be whole integers. And most of these are 5, 10, 9, 14. Suddenly we get some weird duplicates. There's a 3, and then there's a 3.1. Let's highlight these two rows for a moment. Nucleus 3 has a different volume to nucleus 3.1. And indeed all the primary fields between them are different. But let's scroll across to the secondary fields. And we realize that the secondary volume for both of these two cells is identical, as are all of the secondary fields, and indeed all the tertiary fields. This naming convention in the labels in single cell mode tells us that we have found multiple nuclei inside of the same cell. FOCUST appends an index to the number of nuclei that were found inside that cell. So in this case, nucleus 3 was found inside of cell 3. Well, I should say there was a nucleus found inside of cell 3. And then we found another one, so we called it 3.1. And if we found another one, it would be 3.2, and so on. So this flag of a decimal point here tells us that there were multiple nuclei found inside of that particular cell. We can see that it's happened in a few places. The reason we chose to give this sort of information to the user in this way is that It could either represent a biologically interesting phenomena that you're trying to investigate. Bi-multinucleated cells are a thing, but it could also represent an error in segmentation. Perhaps your nuclei are being split into too many pieces, and that's something that you can then visually see in your data and go back and correct and rerun your analysis. So let's have a look. Let's figure out if this particular nucleus, or these two nuclei that are apparently in cell 3, if they are indeed accurate segmentation or if this is an error. So where to look? Well, the labels reflect the cell. So let's have a look at secondary objects. And I'm going to look for cell label 3. As I'm hovering over these cell labels, I'm looking at the toolbar of FIJI to find the value. Value equals the label. So it's going to be up here somewhere. There it is. So this orange colored cell is cell number 3. And our data tells us that we found 2 nuclei inside of cell number 3. Let's go ahead and open our primary objects for single cells 1. And let's go to slice number 12. Aha. If we zoom in on cell number 3, we can see that there are 2 nuclei that would definitely be enveloped inside of cell 3. So our data was right. We have found a cell with 2 nuclei. Again, whether that's biologically relevant or not, we could load up our original image and have another look. But we'll notice our tertiary image will also reflect this discovery in that we have 2 absent nuclei from cell number 3. So we have nucleus number 3 and 3.1 sitting inside this space. But if we have a look at the primary objects again, and we have a look at their values by hovering over them and looking in the toolbar, we can see that this nucleus is value 20, and this nucleus is 27. So something's wrong. In order to give labels names with decimal points and fractional parts of numbers to them, you can't use standard ImageJ approaches. So we actually created a new image type that allows us to save image labels as floats or really long large numbers. So if we close our secondary objects, that brings us to the last output of this experimental run, which are these primary objects relabeled single cells. When we generate our single cell outputs, we have some standard primary objects, some standard secondary objects, and some standard tertiary objects, and they all have unique labels. It's important for data integrity that we don't muck around with those original primary objects. So we create a second version of this image where any duplicate nuclei are renamed. You'll notice that instead of a 16-bit image, this is actually a 32-bit image. That means that we can save float numbers as object values. So if we zoom into both nuclei here, and again, this top nucleus, looking at the toolbar, is value 20. This one is 27. Now this is value 3 and 3.1, which nicely aligns with the fact that they both were found inside of cell number 3. So FOCUST automatically pairs nuclei with cells in 3-dimensional space when we're running single cell mode, and it gives you a record of that discovery by appending indices to the label names. This has been incredibly useful for our research. It allows us to really differentiate whether we have good segmentation or an interesting stem cell phenomena that we need to investigate. So in summary of this particular discovery here, we have batch process friendly data with matching nuclei to cell to cytoplasm, where a single row represents a nucleus and its parent cell and various components therein. Thank you.